Okay. It's not really imperative if it doesn't work. It's
That's
fine.
pretty good.
Can you see that okay?
Yeah.
Yeah.
I'm not blinding you, am I?
Okay. Hello, I'm Kristen. I work at the reptile zoo. You might have seen me earlier today. Did you guys grab me a marker? Oh, you did.
Yeah,
You're so wonderful. Thank you. Okay. So originally, I think I said the title of this was going to be Kentucky Venoms or Venoms of Kentucky or something. And then I kind of got into it a little bit. So I don't know about the rest of you, but when I began learning about venoms, and as I continue to learn about venoms, a lot of times you kind of read something and you're like, what? So I thought maybe instead of trying to talk about like specific snakes here, I would just break it down a little bit further and go back some, because I think probably everyone is confused. And so this is going to be my attempt to explain a couple of components in snake venom, kind of what they are and how they work, what they do in a normal situation not related to snake venom, and like what they do in a bite, and maybe how they can be useful. So I've just picked a couple. Snake venom is really complex. There are many, many different compounds in any snake's venom. So the ones that I have chosen are common for many different species, including all of the species that are native here in Kentucky. So I've thrown in some pictures of our native guys, just so you have something pretty to look at. And then I'm gonna see if we get confused or if it would help to put stuff up here. Because I feel like a lot of times you look at something and you're like, oh my gosh, there's a lot of science words on there. And even if you think that's cool, you're a little bit like, whew, this is a lot. So that's where we're gonna go with this. If you get confused or you don't know what I'm talking about, let me know, because that's the whole point, is to try to talk about this a little bit. Here's something that I wish I had realized or someone had told me a long time ago, and realized that it like continues to apply throughout everything. When scientists name chemicals, they're just naming them by what they are, like what are the things that are in them and how are they put together. So they're not, at least in a basic sense, naming them on what they're doing initially. If they find one that does something, they might give that one that name, but that's not the name of the entire class. Does that make sense? So a lot of times you hear people, and I'm going to mention different proteins that are in snake venom. That's just what it is. It doesn't really mean any more than that's the components and how it's put together. So I wish that would, I don't know, I somehow didn't realize that initially, so maybe that'll help you. Also, just because something is, as we get into it, like a phospholipase A2, doesn't mean that every phospholipase A2 does the same thing. And just because different chemicals can have different actions, sometimes in a biological sense, the outcome can be the same. For example, you can bleed from lots and lots of different things. And they can have different ways in which they make that happen. So, we're going to talk about a few things. So this is a picture of some copperhead venom. This is divided up into the old subspecies because a lot of researchers still recognize those. So, anyway, many snake venoms are yellow. That's because of this stuff, amino acid oxidase. And what do? They oxidate amino acids. What does that mean? Does anyone know what that means? Yeah, okay. I didn't either. So, that's fine. Basically, if you break it down a little bit further, and I think that's on the next slide. Yes. Okay. So, in any biological system, actually maybe I should say on this page, you have proteins that do things. If there is a biological system in your body, which actually everything in your body is a biological system, there are proteins that make that stuff happen. And this is very broad, but it's kind of intended to be. Like, that's my point. You don't necessarily have to know exactly what every single protein does to know that in general, for something to work in your body, there are proteins involved. And L-amino acid oxidases, of which there are several, of course, they change the structure of different proteins, or they break some proteins down. And by doing that, they cause things to happen. And that's kind of all that you need to know to kind of understand what they do. And then you can apply that to any different sort of system. So like, they can make you have swelling, they can cause bleeding. Remember I said lots of things can cause bleeding, everything can cause bleeding basically. They can cause you to clot, they can cause you to not clot, they can cause you to cell death there, they can cause cells to die, they can also cause cells to talk to each other, they can cause things to move around. So there's lots and lots of almost every system that uses these various chemicals. When you have a bite, the reason that we care about them in a snake bite is because they cause cell death of the cells in blood vessels. So in your body, right, you have all these many blood vessels, and your heart is like down here, right? And some of it's coming out this way, and then it comes back in this way, right? Well, right near your heart you have really big blood vessels, but out near your hand, that's a hand, right?
It's not my hand.
This is Jim's hand.
Hey, I have a thumb.
Oh, wait. All of these blood vessels are very small, right? They're capillaries. Actually, they're really tiny. I can't even draw them that small. What happens when the snake venom gets there in this particular instance is the venom goes to this spot, goes to the capillary, and it actually causes some of those cells that are making up the actual blood vessel to die. Well, when that happens, what do you think happens to all the stuff inside the blood vessel? What's it going to do? It comes out. It leaks. And then you have all that stuff coming out into your hand or wherever the snake bit you, and it swells, right? And it probably hurts some. And you can start to have things that are in your interstitial space, so the places between your cells, just kind of in the where there isn't an organ, and that can cause problems for that to be there. So, I mean, I realize this is a terrible cartoon, but the point is you have direct damage to the cells in this particular case, all right? And one thing to understand about their action is when LAAO breaks this protein, there is a byproduct. Almost all chemical reactions, you have 2 or 3 things, and then you put them together, and then you get 2 or 3 other things. Well, when you have this particular reaction with LAAOs, one of the byproducts is hydrogen peroxide. How many people have ever put hydrogen peroxide on a little bit of spilled blood to clean it up? Anybody done that? What does it do? Fizzes, right? So, it's not exactly what happens when it's inside your body. But because of that, there are potential therapeutic applications for these because of that byproduct of hydrogen peroxide. And they're actually used in living systems sometimes too as part of the immune system, because they create this hydrogen peroxide, and then that can actually break the cell membranes of bacteria, viruses. I didn't know this, but I have on there anti-leishmaniasis, so also some parasites. And that's something that can be obviously very, very useful. And then here's another thing along with bleeding, or kind of goes hand in hand with bleeding. They also can cause aggregation of platelets, so that's forming a clot, and they can also cause platelets to fall apart. All right, has anyone ever heard of, Kat, do you want to say it?
snake venom metalloproteinases.
Cat just likes this word.
Yes.
So they are so called. The metal part of their name. Wow, this is really great. The metal in the name is because these contain at least one zinc molecule. And in general, in the body, they function by breaking proteins. And didn't we just talk about that? Well, yes, we did. This is just using some slightly different mechanism. You don't have to know exactly what it is to at least understand that they break down some proteins. And they're involved in a lot of the structures that are in the cell membrane that cause cells to stick to each other, or to not stick to each other, or to talk to each other, or to receive signals from other places in the body to tell them to do something or to stop doing something. So that's kind of why they exist in general. And in a bite, they kind of mess all that signaling up, is basically the way to do it. So the last one we talked about actually kills the cells in the blood vessel. In these guys, when you have like one cell, here's the nucleus, all right? And this actually causes some of the places in the membrane they bind to the toxin, and then the cell itself gets leaky. This is a really great diagram, I know. But that can cause bleeding because it's damaging the blood vessel. But it also can cause, because these various parts of the cell are leaking out and becoming loose in the tissue, that can cause swelling, inflammation, pain. Again, this is similar, right, to LAAOs. One thing that I did not know about, which is how these are studied in the actual, biological systems now, and I don't know, Kat, you have to tell me if you knew this, but in human systems and in many animal systems, metalloproteinases and disintegrins are found together. Did you know that?
No.
Yeah, it's like a system. They're called atoms. Anyway, I know, mind blown. And in snake venom, when the venom is injected, the metalloproteinases partially break apart, and the disintegrin is the one section of the metalloproteinase. So, what do? Well, just like I was saying a minute ago, they allow cells to talk to each other, to stick together, to break apart, all of that sort of thing where they're interacting with each other in some way. And they are extremely common in the body. You have many of these things in your body. They're doing all sorts of normal, everyday stuff that your body has to do. They're digesting food, they're sending signals to other cells, they're regulating your hormones, all of that sort of stuff. And when a snake bites you, and you have all of these things injected into you, they get in the way of those normal processes. So instead of your cell talking to your own disintegrin, which is telling your cell to do something it's supposed to do, then the snake venom disintegrin gets in there and talks to your cell instead. And it can tell it to do things that you don't really want to happen. But they're also really interesting therapeutically. And this is one of the groups that's actually been extremely well studied and has a lot of different compounds that are of interest. So the first one I just picked these two because they're at least sort of native. It's not our pygmy rattlesnake, but it is a pygmy rattlesnake that has this disintegrin called barberin. And that is the, I guess, template, but that's what they've based this drug on. I don't know how to say that. Epitaphabetide? I don't know.
There you go.
And that is something that's actually made by several drug manufacturers. They don't make it from the snakes anymore. That's modeled after this disintegrant in the snake venom. But it actually prevents platelets from aggregating, so it prevents the clot from forming. And the nice thing about it is it's actually controllable and dose dependent. So if someone's having a specific type of heart attack where they have a blood clot forming, you can give them a very specific amount of it to control how much ability their blood has to clot. And then you can actually cause it to be reversed as well, which obviously you want to have that happen, because otherwise the person would just die of something else. So that wouldn't be very good. And then many of you probably know that copperhead venom has been used in research on cancers. And so this is actually a disintegrant in copperhead venom that is the primary source of all that. When we say like copperhead venom is used in breast cancer research, it's this disintegrant called contortrostatin that has been used. And what it does, remember I said that disintegrants a lot of times cause cells to stick together or not stick together. When a tumor forms, it grows really fast, right? And then it sends out bits of itself to other parts of the body. That's what metastatic cancer is. Then they go somewhere else and you get a new tumor forming somewhere else. Well, contortrostatin actually helps prevent that spread from happening. And so it prevents the communication between the cells that say, hey, go out and find a new place to live. That doesn't happen with it. So that there's not a drug being made currently from contortrostatin, but just recently, within the last couple of years, there's a different disintegrant in the venom of saw-scaled vipers. And they are taking the two disintegrants and actually putting them together in the lab and making one that's a combination of the two. And that's actually what's being used currently in research in the same direction towards trying to prevent growth of cancer. So I didn't know that they were doing that combination thing. I actually just learned that in looking this stuff up. So I thought that was pretty cool. All right, this is the last one I'm going to talk about. So these are phospholipase A2. Phospholipase just means it breaks a fatty acid, and it does it in one specific location. And again, a lot of these things probably seem the same. They do kind of the same thing. That's because biological systems are redundant. And so there's many things that cause similar reactions, and also because we're all related to each other. So the same thing that causes digestion in a snake causes digestion in people, and that means it can be disrupted by venom as well. But when you change fatty acids, especially if they're in a cell membrane, it can change what that cell is doing, what that cell is receptive to hearing or moving to or whatever. In a bite, they really have a lot to do with pain and swelling. But they also have this really interesting thing where they can be neurotoxic, they can be myotoxic, or they can be both. Some of you who were at the zoo today may have heard Jim talking about his bite that he took from one of the South American rattlesnakes. And in that bite, he had neurotoxic symptoms.
It was an accident. I didn't take it.
Remember what I drew. He had neurotoxic symptoms. He had the inability to open his eyes. He has respiratory paralysis. He had severe neurological symptoms, but he also had myotoxic effects. He also had a lot of muscle degradation products going through his kidneys. They had to give him a whole lot of fluid to try to combat that. And so that was a specific PLA2 that is known to exist in that snake's venom that is both neurotoxic and myotoxic. And when you have something like that that can be really targeted, that can be useful in research as well. So if you have a PLA2 that normally is involved in whatever, any biological system, if you have a snake venom PLA2 that affects that system, then you can sometimes learn how to disrupt that system or how to fix it if it's disrupted by some sort of disease or problem. And so a lot of them are used in that sort of technique to try to figure out, like, where can we use this or put this in order to try to make it more useful. I think this is my last slide.
You
I was
think?
trying to make this be relatively quick. Yes. Yes.
And not painful.
Right. So, I don't know if you feel like you learned anything. I was trying to make it simple, but also kind of explain it. So I don't know if I did a good job or not. But if you have questions or you want to talk about it some more afterwards, just find me. I'm easy to find. I'll be around, and I'll be at the zoo tomorrow. And I will get out of the way so Kat can get out the live animals, which I know everybody really wants to see. So, thank you very much.